Hello everyone, this is Dr. Karaki. The topic of this presentation is amino acids and proteins. Proteins are the primary structural and functional polymers in living systems. They are large, complex molecules that play many critical roles in the body. They have a broad range of activities, including catalysis of metabolic reactions and transport of vitamins, minerals, oxygen, and fuels. Some proteins make up the structure of tissues, while others function in nerve transmission, muscle contraction, and others. Proteins are synthesized as a sequence of amino acids linked together by a peptide bond or amide bond. Over 300 amino acids present in nature, only 20 amino acids are engaged in the protein synthesis. Each amino acid has a central carbon, called the alpha carbon, to which 4 different groups are attached: a hydrogen atom, an acidic carboxyl group, a basic amino group, and a side chain R, which is different from one amino acid to another. This structure is common to all amino acids but the proline, which is a cyclic amino acid. All amino acids contain at least one asymmetric carbon, except for glycine. Look, for example, at the structure of leucine, and precisely at the carbon with the red star. This alpha carbon is surrounded by 4 different groups. Let me show you. You have the carboxyl group, the hydrogen atom, the amino group, and this bulky group. So, this alpha carbon is surrounded by 4 different groups, making leucine a chiral molecule. Now look at the structure of glycine. It's an achiral molecule. Why? Because it doesn't have 4 different groups. It has 2 hydrogen. Okay? This chirality gives 2 isomers that are optically active, which means they can rotate plane polarized light. These stereoisomers, also called enantiomers, are non-superimposable mirror images, and they are like the left and the right hands. There are 2 configurations of amino acid, the D and the L configuration. D stands for dextro, or right. It's when the amino group is headed to the right. L stands for levo, or left. It's when the amino group is headed to the left. Now let's talk about the classification of amino acids. Based on their chemical structures, amino acids are either polar or non-polar. The polar amino acids with hydrophilic residues are exposed to the surface of the protein and they love water, whereas non-polar or hydrophobic amino acids, they are usually buried in the hydrophobic core of the protein and they are out of contact with water. Now let's start by naming the aliphatic non-polar amino acids. We have the glycine, alanine, valine, leucine, and isoleucine. These amino acids have saturated hydrocarbons as side chain. Although glycine has only a hydrogen side chain, it's also included in this group. Now for the aromatic non-polar amino acids, we have the phenylalanine, the tyrosine, and the tryptophan. These amino acids absorb light at 280 nanometers, and tryptophan had the highest absorption due to its 2 aromatic rings. The third group of non-polar amino acids are the ones containing a sulfur atom, such as methionine and cysteine. Methionine has a methyl thioether group in its side chain, whereas cysteine have a thiol group. Cysteine plays an important role in stabilization of protein structure because it can participate in the formation of a disulfide bond known as SS bridge with other cysteine residues, cross-linking protein chains and thus stabilizing the protein structure. Now the polar amino acids are divided into 2 groups, the neutral and the charged amino acids. In this slide we'll talk about the neutral amino acid. Among them we have the asparagine and glutamine, and the hydroxyl containing amino acids serine and threonine. As for the charged amino acids, some are positively charged such as lysine, arginine, histidine. Others are negatively charged amino acids such as aspartate and glutamate. This slide contains the abbreviation list of the 20 amino acids. You can pause the video to memorize them. Now it's a quiz time. After memorizing the 3 letters and 1 letter abbreviations of amino acid, test yourself with the following quizzes. In my next video, I'll be talking about the determination of the charge of amino acids at different pH and how to determine the isoelectric point. Thank you for watching! If you like it, please subscribe, like and share.